وين منطقي؟
Fantastic. Okay, well today inshallah we will not have a very long lecture, like a lot of the stuff you know. We will talk about the posterior pituitary hormone. Yeah, how many posterior pituitary hormones do we have?
Two.
Okay, so we have only ADH and oxytocin, and they look like each other chemically. They're both 9 amino acids and very small polypeptides. ADH is mainly synthesized in the supraoptic nuclei, يعني 5/6 of it is synthesized in the supraoptic nuclei, 1/6 is synthesized in the paraventricular, the opposite is 3/4 oxytocin. يعني very easy. You don't have to be in medical school to know that. Now, as usual, whenever, if you really want to understand a hormone, you want to know where it's synthesized, where is it released from, how does it get from where it's released from to where it acts, and when it gets to the site of action, what exactly does it do? That's what we want to know about ADH. If you know that, that's all you need to know about it. Okay, so we know where ADH is synthesized. It's synthesized in the hypothalamus, in the cell bodies of these magnocellular cells. They're a little large, that are located mainly in the supraoptic nucleus. It simply moves down the axon to the nerve terminal. And as ADH is synthesized, another protein called neurophysin is synthesized with it. And as ADH moves down from the soma down to the nerve terminal, it is bound to this protein called neurophysin, even though it's loosely bound. It's loosely bound to this protein. The minute it's released, the two separate. Neurophysin, we don't know exactly what does it do, what its function is. You don't need to worry about that. And ADH, what happens to ADH? It's what?
It will be stored and released from the posterior.
When it's released,
It goes to the kidney.
but how does it go?
Blood.
Yeah, then it's a protein. It's dissolved in the water of the plasma. It doesn't. It's not bound to anything. Remember, steroids and other hormones, they can be bound to plasma proteins. This is a small peptide, so it's water soluble. It simply spreads in the extracellular fluid. Its concentration in the extracellular fluid will increase. It will go to the site of action, and the site of action of ADH are the luminal cells. Its site of action are the cells that line the nephron at the late distal tubule, cortical collecting duct, and medullary collecting duct. Period. These three areas of the nephron are not permeable to water except in the presence of ADH. Okay, so what else do we want to know about ADH? What stimulates the release of ADH? Increased osmolality. It's not dehydration, it's increased osmolality, which is totally different. The number one stimulant of ADH is an increase in osmolality. If you increase the osmolality by only 1%, ADH will be released. So this is, and how do we know that? There are some experiments we showed you when we talked about the hypothalamus, but we will tell you about it again. If you increase the osmolality in the supraoptic nuclei of rats and measure the action potentials, you will see a significant increase in action potentials. And that's how these cells release ADH. They're stimulated, an action potential is created, and the nerve terminal, instead of stimulating another neuron at the synapse, it simply releases ADH. And of course, this is a process of exocytosis, so it requires calcium, or it requires ATP, like all exocytosis mechanisms. When released, circulates, it goes to the tubular cells lining the late distal tubule, cortical collecting duct, as well as medullary collecting duct. What does it do when it gets there? It's by a process that involves cyclic AMP, second messenger. And remember, this is a protein. The first lecture we talked about how proteins function. They usually interact with a receptor that's usually located in the cell membrane, and that does a number of things, leading to the increase in cyclic AMP. An increase in cyclic AMP in these cells promote the synthesis of a protein called aquaporin 2
not only does it promote its synthesis, but it moves it from the cytoplasm to the luminal side of the cell membrane, and it's inserted there. This protein, aquaporin 2, is highly permeable to water, so this is really what we call water channel, and a lot of water then start going into the cell. There are two other aquaporins on the basal side of the cell, on the basolateral. They're aquaporin 3 and 4, but apparently they are not affected by ADH. Okay? So that's how it functions. Now, what do we know of other stimuli that increase the release of ADH?
Yes.
We know of two, yes, I mean,
يمكن reduce blood pressure.
yeah, reduced blood pressure, that's for sure, would cause an increase in ADH. And decrease in blood volume. Okay? However, ADH release is not very sensitive to those, as not, it is sensitive, but not as sensitive as the change in osmolality. And remember, ADH is also called arginine vasopressin. Vasopressin, you know what that means, and it causes vasoconstriction. And when ADH was discovered, we found out that large amounts of it cause very strong vasoconstriction. So for a short period of time, we thought it plays an important role in the regulation of blood pressure. It plays a role in the regulation of blood pressure, but its number one function is really to regulate osmolality. Well now, how do we know its number one function is to regulate osmolality? لأنه if you change osmolality by only 1 milliosmol, you will have a response. If it's going up, ADH will increase. If it's going down, ADH will decrease. But for blood pressure, you have to reduce the blood pressure a little bit more than 1% to have ADH release. And for blood volume, yani if somebody has a hemorrhage, they have to lose about 10% of their blood volume before we see an increase in ADH. Well, do we lose 10% of our blood volume on a daily basis? No, sometimes, no, not so much. It happens once, and you cannot lose 10% every day. So if somebody has an accident, their blood a lot, at one point their ADH will start increasing after they lose about 10% of their blood volume. So now, from these two pieces of information, you know, from day to day, today, the number one role of ADH is to regulate osmolality. But it also, if you happen to lose 10% of your blood, it will go up. What does it do? The same thing, it increases water reabsorption, and you lost a lot of blood, so you want to replace that amount that you lost. Any questions about ADH? Any questions طبعا everything is clear? Okay, well that's كلها الصور, you read it at home. We said all this. Let's read it. And this is an important. This one here is the relationship between ADH and change in osmolality. And you can see, I mean, it's almost. Of course, a straight line with the 45 angle with the x and y axis, and it's a very clear relationship, directly proportional. You change the osmolality only by 1%, you will have a response in ADH. But look at the loss of blood, change in blood volume. You have to decrease blood volume by about 10% before you start really seeing a steep increase in ADH. So you have two figures here for two different things. One, the relationship between ADH and osmolality, and one between ADH and blood volume. And you can see this responds a lot faster. It changed, like I said, with only 1% in osmolality will cause a change in ADH. But you need to lose about 10% of blood, and right here, before you really start seeing an increase in ADH in relation to even losing more blood. Kind of the same thing goes for blood pressure. I mean, it doesn't respond immediately, but it responds probably